proceed to what? The sequence of events in acute inflammation. Sequence of events. So there are just two sequences that are going to happen during an inflammatory what? Response, or during an acute inflammatory response. What is that? There are two. There are what we call vascular changes and what is cellular changes. Very, very important. And cellular changes, they are further categorized into what? Leukocyte recruitment, and the other one that we'll talk about in the other video, which is the leukocyte activation. Okay. So now, before we go to vascular changes, we need to understand something, because this can't happen minus what? The stimuli, or minus the cause. These causes that we talked about, what do they do? So let's try to imagine to say this is a cell, okay? This is a cell. Like, let's just pick a white blood cell. Let's just pick the famous one, right? Which is a neutrophil. Or let's just pick macrophages. Okay, let's just pick macrophages. Or let's just pick neutrophils. I like neutrophils. So this is a neutrophil. Okay. So now we've picked a neutrophil. Now, how is neutrophil going to be activated and lead to what is vascular changes and what is cellular changes? Cellular changes. How does that happen? And the question is, so the question that was being asked long time ago was that how does the immune cells like neutrophils or these white blood cells differentiate the normal tissues from foreign invaders? It is because of what? They've got receptors. So they've got specific receptors for what? Foreign invaders. If a foreign invader comes in, they're going to be recognized by what? Those receptors on the white blood cells. If they are recognized, the white blood cells is going to be what? Activated, and they activate the inflammatory response. And what are those receptors called? They are called pattern recognition receptors, which are also abbreviated as what? PRR. So what are we saying? So neutrophils, in order to be activated or to recognize these foreign invaders, maybe infections, they have what we call PRR. So they have what we call PRR, pattern recognition receptors. They use these pattern recognition receptors to recognize what? The foreign invaders, or the cause of what? Inflammatory response. Okay. Now, what are these PRR? Do we know the types of PRR? There are two basically major types. There are two basically major types of pattern recognition what? Receptors. The first one are what? Toll-like what? Receptors. So as you can see, this one. The first one is what? Toll-like what? Receptors. So what is the first one? So the first one is what? Toll-like receptors. So this is the first good example. So the first good example is toll-like receptors. Is this one that is embedded on the what? Cell membrane, and it is also inside the endosome, inside the cell. Okay. So this is the toll-like receptors, which we will also describe. Okay. What is the other one? The other one is what? This one. This one is what we call inflammasome. So this is what we call inflammasome. Okay, so these pattern recognition receptors, they are of two types. There is the toll-like receptors and also the inflammasome. Toll-like receptors, they are like in our body. They've been discovered about what? They're about 10 members. Okay, there are about 10 members of toll-like receptors. And inflammasome, okay, basically this one is just it's a lot, but it's basically it has to be one group. Okay. Now, if you talk about the toll-like receptors, so the toll-like receptors now, where can you find them? You can find them on the what? Cell membrane, and you can also find them in this structure which we are calling what? The endosome. Okay. So where can you find the toll-like receptors? You can find them on the cytoplasmic membrane or cell membrane, and also in the endosome, on the endosomic membrane as well. Okay, this is what we call the toll-like receptors. So toll-like receptors, how do they respond? So they respond to a lot of what type of microorganism. Basically, these all infection that we talked about here, they're going to respond to bacterial, viral, fungal, parasitic infection as well. Okay, they're going to respond to that. The moment they respond, Okay, as well as this one, endosome also you're going to have like, if you look at the neutrophil, they undergo a certain process which we call phagocytosis. We'll come and talk about it. If they engulf something, then they're going to have viral DNA inside the endosome too. Okay, so here there will be what? A viral DNA. Okay, here there will be a lot. So viral DNA, some proteins from the what? Carbohydrates from those bacteria. They are going also to activate the what? This receptor. And also from outside here, the bacteria, those bacterial products, the bacteria itself, they're going to activate the toll like what? A receptor. Okay, those are the things that are going to activate the toll like receptors. Okay, I'll talk about what happens if now these are activated. They're going to release something important. Okay, let's go to another one. This one, as you can see, it is within the cytoplasm of this neutrophil, and we call it what? An inflammasome. So inflammasome now is some. Remember that from the definition of an acute inflammatory response, we said to say this is a type of response that will respond to both what? Foreign invaders and what? Necrotic tissues. Now, what do you think necrotic tissues are going to be recognized? They're going to be recognized by what? Inflammasome. Okay, inflammasome, they're going to respond due to what? Necrotic tissue substances. Like what? If a cell dies, remember that if a cell dies by necrosis, they're going to release their cellular contents. If they release their cellular contents, like what? ATP, they're going to release crystals, a lot of stuff. So they're going to release what? ATP, adenosine triphosphate, they're going to release ATP, okay? Then also, they're going to release what? They're going to release crystals, okay? They're going to release a lot of things, okay? Now, if you activate the inflammasome, because of that, this inflammasome is going to be what? Activated as well. So now let's start with this one. What's going to happen to this? So if inflammasome is activated, there's an enzyme that's going to be activated as well. We call it what? Caspase 1. We call it caspase what? 1. This caspase 1 is going to activate a very, very inflammatory cytokine that we need to understand is what? The famous one is interleukin 1 beta. Okay? It's going to activate what? Interleukin what? Interleukin 1 beta. Okay? Interleukin what? 1 beta. So it's going to activate that one. So let's just say interleukin 1. So this interleukin 1 is one of the important cytokine that's going to be one of the triggers of what? Inflammatory response. So let's just put it here. Okay? So if inflammasome is going to be activated due to those processes that I've talked about, they are going to release what? Interleukin 1. And what about this one? Toll-like receptors, if they are activated, they are going to initiate the signal transduction pathway that will lead to what? Translation of proteins and also production of what we call cytokines. So the end product of activation toll-like receptors, they are going to start producing what? Cytokines. So they are going to start producing what? Cytokines. Okay? Cytokines like what? So cytokines, we will talk about the famous ones or the major ones. Tumor necrosis factor, okay? Alpha. Tumor necrosis factor, alpha. Also, remember that we also had interleukin what? 1 from inflammasome there, okay? There is also interleukin 6 that's going to be produced because of toll-like receptor activation. There are a lot of things that are going to be activated as well, or they are produced. There is also what we call histamines. Histamines, they are going to be produced. Remember that these inflammatory mediators, we'll come and talk about them, the chemical mediators of inflammation. We'll come and talk about them, the histamines, the bradykinin, we'll talk about them. So here, we just have to know that after maybe a trigger, this cell was exposed to what? An infectious agent, a foreign invader, maybe the necrotic tissue, they're going to be activated. These receptors are going to be activated and produce what? Cytokines. Cytokines like what? Tumor necrosis factor alpha, interleukin 6, interleukin 1. Then what are they going to do this? They are going to now start changing they are going to initiate the vascular changes. So you need to understand that. Now, vascular changes. Let's talk about the vascular changes. What are the vascular changes? Vascular changes basically are of two types. So there is what we call vascular dilation, and also what we call vascular permeability. So what are the two important vascular changes that are going to happen? There will be what? Vascular dilatation, okay? dilatation or dilation. Okay, what else? There's also what we call vascular what? Permeability. There will be increase in the what? Vascular permeability. Okay, increase in what? Vascular permeability, and the increase in what? Dilatation. And again, we need to understand what can lead to what? Dilatation. So the first thing that will happen is this. So before dilatation, there will be what? Vasoconstriction of what? These blood vessels. After vasoconstriction, it's just transit within some seconds, then it will change into what? Dilatation due to this effect. So histamines, cytokines like that, bradykinin, they'll cause increase in the dilatation and also increase in the what? Permeability. Now, what are the effects if the blood vessels are what? Dilated? You need to understand the effect if the blood vessels are what? Dilated. If they are dilated like that, so they become like that. So they are dilated like that. The lumen is becoming large like that. What's going to happen? What is the normal flow of blood in blood vessels? It is what? Laminar type of what? Flow. Now, if there is dilatation now, that laminar normal flow is going to be what? Distorted, and it will be changed into what? Into the stasis type of what? Flow. So due to this dilatation, what's going to happen to the blood flow? There will be what? Stasis of what? Blood. Very, very important concept, okay? And if there is stasis of blood, basically there will be increased blood flow to that type, to that injured blood vessel, to that activated blood vessel. If there is increased blood there, then what's going to happen? There will be now redness. Redness, basically it will show that there is increased blood flow to that what? Capillary, or to that blood vessel. And also there will be what? Heat, because of what? Increased blood flow to that what? Tissue. Okay, yeah. So since we've talked about two, now what is the other thing? The other thing that's going to happen. Now let's talk about what are the things that are going to increase what? Vascular permeability. So stasis, we've talked about it. What about vascular permeability? What are the things that are going to lead to the increase in the vascular permeability? What are those things? The first one is what? The first one is endothelial. The first one is what? Endothelial cells contraction. Okay. As you can see, these smaller cells here, these are what we call endothelial what? Endothelial cells. So these cells are called what? Endothelial cells. Endothelial cells, these are the cells that are going to line all blood vessels. Okay. Now, what's going to happen? For increasing the permeability of the blood vessels, these endothelial cells, they are going to start contracting. So each endothelial cell is going to contract. So let's try to imagine this one. So this one, if it contracts going like this, also the other one going like that, it's going to leave a certain space there. Okay, if it leaves a certain space, that space between two endothelial cells or two or more endothelial cells basically is going to lead to what? Increasing the permeability of what? Endothelial cells. So that's what it means. That's the first mechanism that's going to lead to what? Increasing the permeability of what? Endothelial cells. What is the other one? The other way is what? Endothelial damage. So endothelial what? Damage. So direct endothelial what? Damage. So what if you would just have an injurious substance, or the cause of that acute inflammatory inflammation? Maybe let's just pick one, which is maybe thermal injury. Okay, thermal injury can direct damage the endothelial lining of the what? Blood vessel, leading to what? Now leaving a space. Remember that if this is an endothelial cell, then there's just what? Damage here because of heat. Then there will be what? There will be a space between two or more endothelial cells. Basically, that is increasing the permeability of what? Endothelial cells. Okay. What else? Okay. What else? Also, apart from that, okay. Apart from that, there could be there could be other things, like there are some other things like formation of pores, like in the end venues. So formation of pores that can induce a process called transcytosis, but basically that's not a very, very important point, where proteins are going to what? To start going through those pores, okay, or through those vesicles from one endothelial cells to the interstitial what? Space. That is what we call transcytosis. Okay. So that is also a very important process in the increment in the permeability of what? blood vessels. Okay, so we're also talking about transcytosis process. Transcytosis process. Okay, so yeah, so those are the things. Now let's talk about something, because we've got other cardinal signs that we haven't talked about. So these now, because of increasing the permeability of that, what do you think? What do you think? If you look at here, there are also some proteins that are within the blood vessels. Okay, there are some proteins that are within the blood vessels. Basically, let's talk about albumin. Albumin is the major protein that can be found in the what? Blood vessels. If there is increased permeability, what's going to happen to those proteins? Those proteins will start what? Coming out of the blood vessels. Okay, they'll start passing through this space that was left, going to the what? Interstitial what? Space. So these are proteins that are outside here. Okay, what else apart from proteins? Remember that there is also plasma here. There is fluid here. The fluid will start also following the proteins like that. Okay, yeah, so basically what I'm saying is that if there is increase in the permeability of those vascular blood, those blood vessels, there will be escape of proteins from blood vessels, as well as what? Fluid, causing what? Edema, or what we call swelling. Okay, so it will lead to just edema that you see under the cardinal sign. Okay, so what else? And also there is also loss of functional LISA. So functional LISA basically now, if let's say there will be what? There is pain there. If there is pain, basically because of activation of pain fibers that are surrounding where there is an acute inflammatory response, there is pain in there. If there is pain, basically someone is unable to use what? To use the affected maybe let's say the affected limb like that. Okay, that is loss of function. Sometimes maybe the cells that are there, they are severe what? Fibrotic, and what? They are unable to perform their what? Normal function. So that is under loss of what? Function. It's a lot to grasp it like that. Okay, so that's under vascular changes. That's what will happen under vascular changes. That's two things: dilatation and what? Increase in the permeability. Okay, so now let's move on to another thing that's going to happen: cellular changes. Okay, cellular changes, I said in the earlier on, we said we'll just talk about what? Leukocyte what? Recruitment. That, so then leukocyte activation, we'll talk about that because it is more detailed. We'll talk about the phagocytosis process, we'll talk about a lot. Okay, so cellular changes, what does it mean? Cellular changes, it means that now we're talking about cells now. Those white blood cells are the one now that are now involved in the what? In the inflammatory response. Here we just talked about the blood vessel, dilatation of blood vessel, and increase in the permeability. Cellular changes, talking about what? Leukocyte. No wonder we are saying leukocyte recruitment and the leukocyte what? Activation. Okay, so under leukocyte recruitment, there are 4 important things that you need to understand. What are those 4 important things that you need to understand? The first process that's going to happen is what? Margination and what? Rolling. Okay, margination and rolling. What else? The other process that's going to happen is what? Radiation. Okay, the other process that's going to happen is which is diapdesis. The last one is what? Migration, or yeah, migration, which is chemotaxis. Okay, migration towards the infection site or inflammatory site. So basically, these are the things that's going to happen. Okay, margination and rolling, radiation, diapdesis, migration towards the what? Inflammatory site. So basically, what we need to understand is that we need to understand step by step margination and rolling, what it means, radiation, diapdesis, migration toward the inflammatory site. Because if you have been asked about such a question, you need to describe in detail, because there was a question that was being asked. You are going to be examining something like that. Okay, you are going to be examining. Okay, now let's start with what? Margination and what? Rolling. So let me just make more space here so that you can understand fully. Okay, so margination and rolling. Okay, so okay, I'll just redraw that one so that you can see here. Okay. Okay. If that is the endothelial line of the blood vessel, okay, this is the endothelial line of the blood vessel, and this is the blood vessel inside. So this is the blood vessel inside, and this is the what? Interstitial what? Space. Interstitial what? Space here, outside the blood vessel there. Okay. So now let's start with margination. Margination, how does it happen? Okay. So this is a blood vessel. Blood vessel. So now, how does it happen? Remember that here we said initially we said there was stasis due to the dilatation. There was what? Stasis. That is the first thing that was happened. There was what? Stasis. If there was stasis now, remember that if there is stasis, there are some smaller cells that are like red blood cells. These are red blood cells like that. Smaller blood cells, like red blood cells, they will be what? Moving toward the what? The center like that. But larger blood cells like what? White blood cells now, they will be what? Going to the periphery. The reason why white blood cells or leukocytes like neutrophils, they are going to start undergoing margination is because of what? There is stasis. Stasis will make smaller blood vessels to start moving through the center, and the bigger ones, which is the white blood cells, they will start going to the margins of the what? Blood vessels. Basically, they will start being attached with the what? Endothelial cells. So what is this? This is an endothelial what? Cell. So let's try to imagine to say this is a what? This is a leukocyte. Okay? That is a leukocyte. So now, if it goes there, this is margination now. So basically, margination is just the movement of these white blood cells from the center, then going to the margin of the what? In blood vessels there. Okay? Due to stasis. Then rolling. Rolling is because of they are going to start expressing something. Okay? So these endothelial cells, because they were activated, remember that due to those cytokines that we talked about, they are going to start activating these what? Endothelial what? Cells. If they activate these endothelial cells, they will start expressing something that is not normally expressed. If there is no inflammation, they are not going to be expressed. If there is inflammation, they are going to be what? Expressed. What are those? We call them selectins. Okay? They are going to start what? They are going to start expressing what we call selectins. So we are calling them what? Selectins. There are three different types of selectins that are going to be what? Expressed. There is what we call P selectins, there is what we call E selectins, and what? L selectins. Okay? Selectins. So there are P selectins, E selectins, and what? L selectin. These are normally going to be expressed by the what? Endothelial cells. And sometimes they are going to be expressed by what? The platelet what? Cells. Very, very important. Like the P selectin is going to be what? Expressed by the also platelets. Okay? Now if that happens, let's just pick one. If this is a P selectin here, okay? This is a selectin that is there because it's expressed by the endothelial cells, okay? And also this is a selectin. So these white blood cells, they'll start what? Binding to this what? Selectin like that. If they bind, so it's more like this, they bind from one selectin, then they detach again. They again bind to another selectin, either P selectin, selectin, or L selectin. So in that way, they are initiating what we call rolling. Now the question is, what are the parts of the cell membrane on the white blood cell that are going to be attached with the what? The selectin produced by the what? Endothelial what? Cells. What are those? We call them what? These are just sugars, okay? Sugars on white blood cells. We call them what? Cellular, Lewis, okay? So cellular X, Lewis what? Lewis, okay? So cellular Lewis what? Substances that are going to be expressed on the what? White blood cells or on the leukocyte. So basically, these selectins are going to start binding on the what? These sugars. Basically, they are called cellular Lewis what? X like that. Okay? If they bind on that now, they will initiate what? Rolling. So that's where rolling is going to end from. So what else now? The other point is what? Adhesion. So adhesion now, that is where the leukocyte will stop what rolling, and then it's going to what firmly attach to the what endothelial cell, firmly attachment to the what endothelial cell. So now this white blood cell that is rolling, now it will find an adhesion molecule here expressed on the what endothelial what cell here. It will find an what adhesion molecule that's going to be what expressed on the what endothelial cell. Like what? There are a lot of adhesion molecules that are going to be what expressed like what? If you look at here, there is ICAM and VCAM. Okay. So there are a lot of like adhesion molecules that are going to what make sure that the leukocytes start what stops rolling. What is that? We call them what? ICAM. And the other one is what? VCAM. ICAM basically that is intracellular adhesion molecule type 1. VCAM that is what? Vascular cellular adhesion molecule type 1 as well. So VCAM and ICAM. Remember that now VCAM, this cell, this white blood cell will come here. If it finds the VCAM now, it's going to firmly attach to the what? This endothelial cell. It will stop what? Rolling. Now, again, you need to ask yourself, there are some substances that are going to attach ICAM from the endothelial cell to the what? To the white blood cell. We call it what? Leukocyte function what? Antigen. So the first one is what? LFA. Leukocyte function associated antigen type what? Type 1. Okay? This one. Or basically type 4. Sorry. Basically type 4. So this one is found on the what? On the leukocyte. It's the one that's going to attach to the what? ICAM. Sometimes it's referred to as CD11A. Okay? So if this firm attachment happens now, this is what we call adhesion. Then we proceed. Okay? Then you got what? Diapdesis. Okay? So let's call that one diapdesis. So what is that now? Let me write it here. So diapdesis, let's say this is a leukocyte. Okay, this is what we call a neutrophil that is firmly attached to the what? Endothelial cell by the use of what? ICAM or what? The VCAM. Okay. Now this leukocyte now, diapdesis basically it means that this now it will start moving between the what? Endothelial cells. This white blood cells now, it has to move out of this what? This blood, then going into the what? Interstitial what? Space. Okay. So this movement starting from what? Where it was adhered, then it starts what? Moving between the what? The endothelial cells. We call it what? Diapdesis. But diapdesis is only possible if there are also some adhesion molecules that are going to be expressed by the endothelial cells and also the leukocytes themselves. What is that? We call it PCAM. Okay, we call it PCAM. Okay, we call it that's PCAM. Okay, we call it PCAM. So PCAM, that is platelet endothelial cell adhesion molecule. It's going to be like expressed somewhere, something like that. Okay, it's going to help the diapdesis process to occur. This white blood cell here, it will start what? Attaching itself to this what? To this molecule. Okay. Now, if you look at this odd diagram, if you look at any blood vessel is going to have what? The basement what? Membrane. It's going to have the basement membrane. Now, you need to ask yourself, how do these white blood cells pass through this basement membrane? They are going to start releasing some certain what? Enzymes, and we call them what? Collagenases. So collagenases, they will start breaking down the what? The collagen fibers of the basement membrane so that there is a path so that white blood cells can what? Pass from. Okay. So these white blood cells now, since they start releasing those what? Collagenases enzymes. Okay, they'll start eating up this what? Basement membrane, so that now this white blood cell is where is on the what? Interstitial space. Okay, that's where diapdesis is going to what? End from. Okay, diapdesis is what? That's the movement of the white blood cell from adhesion, family adhesion to what? Into this interstitial what? Space by the use of the what? PCAM. Okay. And also by the help of what? Collagenase enzymes that are going to be released by the what? The same neutrophils or white blood cell. Now the last part is what? Migration toward the inflammatory side. Remember that, okay, this white blood cell is under on the what? On the interstitial space. There is an inflammatory, so there is a bacteria here. Okay, there is a foreign invader here. Okay, this is a foreign invader which is here. Okay, which is here, which was the one that was initially the cause of what this whole thing, the vascular changes, the what cellular changes. The one that is here. Okay, it produces something as well. It produces, like, let's say it's bacteria, it's going to produce toxins. Okay, and also remember that there are some white blood cells that are already here, that reside just within the what extracellular space or extracellular matrix. They are already there. Those are the ones that were producing initial what the cytokines, like what histamine, bradykinin, interleukin 1, interleukin 6. This one. So I'm talking about this one. It's the one that is here. It's the one that is fighting against which one? This bacteria. This same bacteria is producing this, and there is a white blood cell that's producing what? Cytokines, a lot of things. Now, these endotoxins and also cytokines, basically the cytokines now that we'll talk about is what? Chemokines, cytokines. So now the cytokines that we're talking about are what? Chemokines. Chemokines, these are just the what? Chemokines, these are just what? Under what? Cytokine. Chemokines, these are the ones that will make sure that this white blood cell that has come out of from the what? Blood vessels, then these are the ones that will recruit what? The white blood cell to the what? To the site of infection, where inflammation is what happening. So it will start doing like that, going like that, okay? Because of what? Chemokines and the endotoxin. This process is what we call what? Chemotaxis. Okay? Chemotaxis is basically the movement of the white blood cells toward the what? The site of inflammation due to the what? Effect of the chemokines. Now the question is, you can ask, how do these white blood cells move from this one place to another? Because they don't have lower limbs like us, where we are able to move from one place to another. So they use what we call pseudopods. So they are going to extend their cytoplasm like that. They are going to extend like this. They are going to extend this cytoplasm like that, one at a time. If they extend like that, they are going to attach that extension of cytoplasm, which we are calling pseudopod, to the extracellular matrix, so that it can pull the whole cell together like that. Again, it will extend another one. In that way, they are going to start what? Moving. Due to the effect of what? Chemokines. So chemokines can be a lot of chemokines, okay? Chemokines like what? So like there are some substances that can promote chemotaxis, like C5 from the complement, okay, and the same one that I talked about, the cytokines and the what? Endotoxins. So that's where the leukocyte recruitment ends from. And in the next video, we'll talk about now if this white blood cell reaches this infection site where there's maybe a bacteria, how does it perform? How is it going to be activated so that it can perform what? Phagocytosis. How does it kill that bacteria, that infectious microorganism, using what? The ROS, which is the reactive oxygen species, nitrogen oxygen species, a lot of stuff. So we're going to talk about it in the next video. Stay tuned and make sure that you like the video and subscribe.